Hello everybody and welcome back. So, we are discussing about the synthesis of the nucleosides. In my previous lecture, I have talked about how the nucleobases, both purines and pyrimidines, can be attached with the sugar moiety, both in ribose sugar as well as the deoxyribose sugar. And then we have been actually discussing a little bit of sugar chemistry itself, how you can do the protection and deprotection chemistry on the sugars, on various positions of the sugars. And we have seen that if you have the ribose sugar, then the C1 prime position you can pretty much protect with any protection group separately, because that has very different reactivity. And then we have also protected the 5 prime with one of the example is with DMT, that is the trital using the triphenyl chloride. And now we have the 3 prime OH and 2 prime OH. Now question is, how can you selectively protect a 2 prime or 3 prime? Now, if you want to protect the 2 prime position, so first thing is 2 prime and 3 prime, these two positions, their reactivity are more or less same, because both are secondary kind of secondary alcohols or secondary hydroxyl groups are present. Now, if you have remember, there are few ways if we want to do the protection, selective protection of the 2 prime position. There are few ways. So, our target is now this is protect, I am writing P1, this is P1 for one kind of protection, this I am keeping DMT on. So, our target is protecting this P2 and keeping this free. So, that is our target molecule now. So, I will quickly go through it and then we will discuss about the nucleic acid synthesis, the final part of it, of this module. So, there are few ways to do this. One of these is if you remember that if you have a diol and you treat this with a carbonyl compound, aldehyde or a ketone, then what they do is they form acetal or ketal. This can be R1, can be R2, it can be aldehyde, it can be ketone. So, this is called acetal or ketal. So, the same thing can be applied in here. In this case, the idea is that first you protect the other parts and then you can protect the C2 position. So, at the beginning, without the DMT, starting point is you have P1, this OH and a free OH. Now, if you treat this is kind of a old method actually with benzaldehyde, then benzaldehyde also forms acetal formation, but more preferably it forms the acetals with 1,3 positions. These two hydroxyl groups are in 1,2 position, 1 carbon 2 carbon. So, those are 1,2 position. If you treat it with benzaldehyde, it prefers to form the acetals in 1,3 positions, which means the 5 prime and the 3 prime will be forming the acetal. So, this will remain open and this will be forming. So, therefore, the 5 prime and 3 prime will be blocked, and now you can do the chemistry on the hydroxyl group in the 2 prime positions. For example, if you want to do TMS chloride, trimethyl silyl chloride, then you will get a OTS, or you want to make a acetylation with acetic anhydride, various protecting groups you can use. So, this will be OTMS, or it can be OAC, and then O, this will still remain as protected, and then you can open this up. So, I am writing you a OP2, this will open up, this will open up. So, you can get a selective protection of on the 2 prime position. So, this is kind of an old technique. What little bit advanced technique is that you use a silyl compound, specifically TIPDS chloride. This is TIPDS chloride. I will show you the structure. The structure is, it is a crowded compound. This is isopropyl, this is isopropyl, so O, this is silyl, diisopropyl and a chloro. So, because of its large structures, this also reacts with a diol, but again 1,2 diol does not work here because they are too crowded. So, here also the preference is 1,3 diol, they will react. So, similar to the benzaldehyde. In this case, so here you will get elimination of chloride, and the C3 you will get the elimination of the chloride. This OH will remain free, O, Si, O, Si, this isopropyl, isopropyl. This one will be forming, this will be forming, and your OH at the 2 prime position will remain free. And after this, you can do the same protecting group chemistry that you have done here. So, it will give you the selective protection as you see this. And later on, you can also open this up just by treating it with a slight amount of acid or any fluoride. And now, if you want to do selectively selective protection of the C3 prime, this group, then what to do? So, here there are some chemistry that have been developed. So, one is that if you start with DMT on, so which means already you have to do 2 steps, one is here protection here, second one is protection there, and if you treat this with TIPS chloride, which is this silyl triisopropyl silyl chloride basically. So, this is also a crowded with high steric crowding silyl compound. So, if you treat this here, obviously there will be nucleophilic substitution. Now, question is which OH will react? This OH can react, this OH can also react. You get both the compounds. One is if you treat it with selective equivalent amount, then this should be O TIPS. This is your DMT, plus you get the 2 prime protection also. TIPS, this can be free in this case. So, either this or either the 3 prime can react or the 2 prime can react. Now, because of this bulky substitution here, the reaction at the 2 prime position is less, much. This is not the major product, this becomes the minor product, because the steric crowding at the 2 prime position is maximum. This is sandwiched between the 3 prime as well as the 1 prime, very close together. So, the accessibility or the nucleophilic substitution by a bulky group is difficult in this position compared to this. So, most often you get a major product for this, but nevertheless you do get a mixture of these 2 compounds. but this becomes a major product and that becomes a minor one. If you really want the 2 prime position to be selectively protected, there are other ways to do it, you can figure them out. You can do individual protection, deprotection, and likewise you can play around with the same protection and deprotection chemistry we have already done here, and then can find out a proper method, but that will require more number of steps, I guess. So, this is one of the ways people nowadays use for selective protections of the 3 prime hydroxyl group. So, now this is actually for the nucleosides synthesis, and we have already done, and this is for the sugar chemistry. Now, today I will come back now to the nucleotide synthesis. So, we have done nucleobases, then we have done nucleosides, that is bases plus sugar, base plus sugar, that is nucleoside, and now comes the nucleotide, base plus sugar plus phosphate. Now, here I will not show individual nucleotide synthesis, we will straight away go for the synthesis of a chain of DNA. So, basically nucleotide synthesis includes, this is one of the key aspect of the chemical biology and enormous contribution from organic chemistry that has enabled us to synthesize short stretches of DNA in the laboratory. So, small fragments of DNA cannot be obtained in the biological systems. In biological systems, all we have are long genes and the chromosomes. To work in your laboratory, to understand the role of specific sequences for biological functions, as well as for, there are plenty of other applications that we will see just in the next module if you have a fragmented DNA. So, synthesizing short stretches of DNA, typically it can be any length, maybe 5 base pairs to up to 50, we can go even longer nowadays, base pairs DNA. They are very important for the working in the biological laboratory or chemistry laboratory. So, we will see now how this DNA can be synthesized in the laboratory, and this is a beautiful chemistry that has been developed by M. Caruthers. Is an American. It is not the same Caruthers who wrote the organic chemistry book. This is Marvin Caruthers, who is famous for the synthesis, for developing a complete protocol for the synthesis of DNA stretches, which is known as oligonucleotides. So, this is basically, when you talk about DNA synthesis, we basically means oligonucleotides. Which means the oligomers of the nucleotides. So, that involves the synthesis of the nucleotides as well. So, the chemistry is a little bit modern organic chemistry and in advanced level. So, I will show you the individual steps just in a moment, but still for this course I am teaching this because in this synthesis lot of different aspects have been developed and they are really interesting. One fact is that this whole of this chemistry is done inside a machine. So, you do not have to go to your laboratory and mix things up in into a round bottom flask or in a test tube and do the chemistry. So, everything is can be performed within in a machine, and that machine is programmed in a with a computer. So, just by putting the software in place, you can comment and then the machine will do every work for you. So, this whole chemistry involves 6-7 different steps that I will show now, but all of those steps happen one by one, step by step, commanded by the computer in this machine. here you can see the machine. So, this chemistry is known as the phosphoramidite chemistry. So, as I mentioned, the organic mechanisms or they are actually a little bit in advanced level, but still I would like to teach it even for the school children, just to show that how chemistry can be done inside a machine, inside an automated machine that can perform by itself. So, this is one of the machine that I am depicting here. The machine is known as DNA synthesizer. This is a typical model of DNA synthesizer, which had bottles here. You can see there are many bottles here that are attached there, and these are the bottles for reagents, the different reagents that are needed in every step. And there are also these are little bit larger bottles, because you need them in milliliter quantity for the synthesis, and then there are smaller ones. This you can see. They are very small actually, for which you need the volume in the level of micromolar quantity. So, we can do very fine chemistry here in the level of microliter volume of material can be, or liquid can be used for the synthesis. These are the other reagent bottles, which are little bit larger, which require a little bit more quantity than this, but much less than this. And these all these bottles are connected to through channels. Here you can see the channels, and those channels go all the way up here, here as well as there. And this is where your synthesis happens actually. So, this is the place where the reaction happens, and these are the vessels. They are called columns or called the cartridge basically. They are the, I will show you what are these. These are basically cartridge which has some solid materials inside. So, these are placed in this chamber, and the machine pumps up all the required reagents in adequate volume all the way to here, and the chemistry happens there. And there is a display over here which can tell you what is happening, which step you are in, or some monitoring stuffs are also there. So, it is a beautiful invention, beautiful discovery that allows you to do multi-steps organic reactions in a very clean manner, all automated. You can simply press the start button in the computer and go home, and the machine will do all the work for you. So, you can, all you have to do to tell the machine the desired sequence of your DNA that you want to synthesize, and then it will do it automatically. So, there are too many machines here. These machines are very much commercially used by the pharmaceutical industries as well as the biochemistry industries, which supplies the oligonucleotides to customers. They synthesize lot of DNA for different purposes which we need for our laboratories. So, we can purchase from them. Many number of machines connected to computer, as you can see. So, all automated. So, now the synthesis here. The synthesis protocol involves a solid phase synthesis, that is what I have written here. DNA synthesis is done in a solid phase phosphoramidite chemistry. Phosphoramidite is the name of the reagent that you use for the nucleobase, or it is kind of a derivative of the nucleobase. You do not have to remember, or you do not have to even understand much of the mechanism that I will show, but still I think it is interesting to see how stepwise chemistry can be done and how clean are they. And the whole chemistry is done not in a solution phase, but in a solid phase. So, the solid powder is packed inside this cartridge. So, if you do solution phase chemistry, why not this is happening in solution? So, if you try to do solution phase chemistry in a round bottom flask in your laboratory or in a test tube. So, for example, let us take a target first. For example, I want to synthesize a random a DNA sequence A T G C, let us say A T G C and A T 5 prime 3 prime. This is my target, that is what I want to synthesize. Now, if you want to synthesize in solution, what you will do first? You will take A, So, this is your A, basically there will be hydroxyl group here, and here you will see you will require a DMT, that you will see a protection would be needed because you do not want to touch here 5 prime. This is your A, plus you have to take your T. Maybe this is OH, or it can be other protection group, whatever chemistry you do. I will show you the problem here. So, and of course, you have to synthesize the phosphate. So, what you need a phosphate and then an X, whatever that can be, it can be a good leaving group. So, because you need a phosphate, I am writing P for phosphate and X for a leaving group. So, the essential chemistry is this will go and X will be eliminated. Let us say this chemistry works, although it is very hard to find do a direct chemistry like this, but let us say this works. So, if you do this chemistry, then what you get? You get a AT sequence. I am not drawing the phosphate. So, A phosphate T basically. In solution phase you get your desired AT. So, first is AT, you get your AT. What else do you get? One A may react with other A, that will give you AA. One T may react with the other T, that will give you TT, plus you will have unreacted T, I am writing hydroxyl group free because of this, plus unreacted A. Not all reactions are complete. So, even if you use when you use these reagents, after the end of the reactions they cannot complete, all of them will not be consumed. So, chances is high that some of these materials will remain as in free form even after the reactions. So, after the very first step, what you get? You get 1, 2, 3, 4, 5 components in your solution. Out of these, only this one you desire. So, if you want to get this in pure form, you have to separate all others, that is again that is a difficulty, or you can move on to the second step, second cycle, and then use G. GOH means I am writing, because since the 5 prime is reacting always, I am writing OH for 5 prime position, GOH. Of course, the G means whole of G, here only the replacement will be. In the second phase, what will happen? You will have A, T, G, your desired compound, plus here A, G, plus T, G, plus G may react with itself, you will have G. You had some unreacted T left over here, that can react now with this G in the second step. So, you will have TG. You had unreacted A, which may again react with G. So, you will have GA, and then again unreacted G will be there. So, after the second steps, you have multiple, so many number of products that are formed. Out of them, only this is your desired compound. So, if you carry on, if you like this, if you want to synthesize just 6 membered ring, 6 membered DNA, then the number of products, side products would be so high that you cannot find even your desired component, or else you will need a purifications in each step, which is really a painful job. So, when you want to synthesize 50 mer long DNA, 50 nucleobases long DNA, then obviously you can see the problem in the solution phase. It is very hard to do in the solution phase. Rather, if you choose to do it in a solid phase is like this. Solid phase synthesis means you have a solid support. In this case, we use glass. So, this is a glass basically, CPG is called, it is a glass basically, glass powder, and that is connected with a functional group. Let us say it is connected with a O, just consider kind of a DMT. Now, this is solid. So, you treat now this with the first nucleobase was A. A, OH, OH. So, instead of I am straight ahead taking OH here, because I have to deprotect DMT, otherwise again would require additional step. So, this is connected, this functional group is connected to the glass solid support. Now, with a proper reagent you can do this reaction, and you can have A here with the OH free at the 3 prime end, and what else you can have? You can have the unreacted of this, but this is now in liquid form. So, after doing the reaction, if you wash away all the liquid and only get the solid, then all you have here is this, plus of course, the glass which did not react, the unreacted glass. And now, I will show when we actually do the cycle of the DNA that we actually make this one inactive, so that it does not react in the further step. Now, in the second cycle, so this is the first cycle, second cycle is you add the T, do the same thing, you get AT, OH free, and of course, all the unreacted liquid form can be washed away. So, if you go like this and obtain your solid all the time, the chances of getting the side products or undesired products, smaller ones, are very, very less. So, the productivity of the synthesis would be much higher if you do the solid phase synthesis, because you are doing it in a located place and you can remove the solid thing out of the reagents, which is in the liquid form. So, that is what the beauty of this synthesis, that this particular phosphoramidite chemistry works on solid support, and it is a solid phase DNA chemistry. So, now if you look at the biology, how these nucleotides are synthesized, how this phosphate is synthesized, how the 5 prime position and the 3 prime position of a nucleobase react. B1 B2 OH. In biological cell, it is a spontaneous process, and this reaction happens in presence of enzyme that we will see in vivid details in the next module. So, how it happens is that it has the triphosphate O, P, P, P. I am not writing the phosphate. This is triphosphate, which is called the, in general they are known as DNTP, deoxyribose, N is nucleobase, T is triphosphate. For example, ATP, deoxyribose ATP is a form of triphosphate. If there is adenine here, it is called adenosine triphosphate. So, in our body we have triphosphates, we have nucleobase triphosphates are there available, and this reaction is catalyzed by the enzyme polymerase. We will see just in the next module. Reacts with the phosphate, eliminates the diphosphate, and that will give you the nucleic acids, phosphate, so B2, B1, OH. That is how you get it in the biological cells. Now, you try to do this reaction in laboratory, you will not succeed. It's very hard to do this reaction without the use of the enzyme, and therefore, we had to develop, and people had to develop for a very long time. There are many different methods that make changes to these variations to make this reaction happen. So, in the solid phase phosphoramidite chemistry, we will see that what is the reagent that we use. Instead of that triphosphate, it is this nucleobase analog that is used. This is the base. Here 5 prime is protected with a DMT, and the 3 prime is protected with this, which has a phosphorus group. And this is because this chemistry works well. It is only for the laboratory, and this after a lot of research and lot of modulations, people have come with the idea that this molecule works best so far. So, here I will show you the first the cycle how it works and then go to the individual steps when you do it in the machine in the DNA synthesizer. So, the first thing that let us start with here, here is your solid support CpG, and you have already done a little bit of synthesis, and now we are trying to add more nucleobases here. So, the first step is that you deprotect the DMT out of this and convert it, this DMT is converted into the free hydroxyl group here, that is the step called deprotection. So, this is attached to the solid glass, and then the reagent, the second nucleobase that you wish to add will pour in, will be pumped into the solid, and then it will react. This OH is free and it can act as a nucleophile. So, this can react with this phosphate. Before that, this molecule is stable, so you need to activate it, and that is done with the activating reagent like this. So, it will activate and then this OH will react to this phosphorus, clipping this out. This is the coupling, means you are joining the solid phase with the new nucleobase. This is coupled together. So, if you turn this as B3, let us say, here is your B3. So, this should be B3. Your B3 is now attached with the solid phase along with the others. So, coupling is done, your essential part is done here. And now, if you see, as I have talked about, that you will have the unreacted material here, the unreacted solid which did not couple will be left over, and if you leave it as it is, then it will react in the next phase. We do not want that. So, therefore, the unreacted thing is capped and gone away. Once that is that unreacted part is capped, it is dead, it cannot react again. So, after that what is left? You get the free form, the same thing without the impurity, without the other solid impurity. Next, if you see here, in your phosphate, the phosphorus is pentavalent and that has a oxidation charge of +5 on the phosphorus. Here if you see that we have started with the +3 oxidation state on the phosphorus, here also you have a +3 oxidation state of phosphorus. So, we need to oxidize this phosphorus further into the +5 state, that is called the oxidation. What it does is that it adds a double bond oxygen here. So, you now have a pentavalent phosphorus, and then you can continue with the cycle. You can still see your phosphorus is not really ready, you still have a short tail, and that we actually can eliminate in the last stage, that I will show. So, I will take the individual step one by one, and then you can see how it happens. So, first let us start with the first nucleobase attached with the glass. This is your CpG, solid, basically solid glass, that is connected via a linker, and that linker is connected with the first sugar. This is the first base, base 1, and 5 prime position is protected with DMT. So, the first step is that you have to eliminate the DMT, and that is done by using a very weak acid, that is trifluoroacetic acid or trichloroacetic acid. Just a little bit of 3% very dilute solution of this, it will eliminate the DMT, will make a free hydroxyl group. Again, you do not have to remember all the mechanisms, but I am just trying to the mechanism is shown here, but I will not explain it in great detail because I want to show you the exact processes first. That is the essence of it. Usually I teach this in advanced level, so that is why I will not explain the mechanism in detail. So, you get a free hydroxyl group here, and your DMT will be eliminated in the form of a carbocation, and this has a beautiful color. This has a orange to purple color, and that is actually a way, I will show you here, to understand whether your reaction is happening or not. Here, once it happens here, you can see the outlet chamber, when the liquid is going out of it, you can see the beautiful orange color that is passing through the channel. So, that proves that your DMT has been deprotected. It is kind of monitoring the reaction actually. So, DMT will be out, and you will have free hydroxyl group. This is the most crucial step actually. And now you have this free hydroxyl group with the solid phase attached, base 1. Now you pump in your phosphoramidite. This is your phosphoramidite. This is base 2 with the DMT protection at the 5' end, and this molecule is pretty stable and therefore is less reactive. So, we have to make it a little bit reactive, so that this hydroxyl group reacts there, acts as a good nucleophile. What we do is we use a little bit of tetrazole. This acts as a catalyst here. What tetrazole does, that tetrazole is just a little bit more acidic compared to this nitrogen. So, this nitrogen group here is little bit acidic compared to this nitrogen, and therefore, it provides a proton here. The proton from the tetrazole is shifted to this nitrogen. Then you have the NH plus in this position, and tetrazole becomes N minus, which can be stabilized by the internal resonance. So, essentially you have this form, and this becomes a good leaving group. So, now the hydroxyl group can react here, eliminating this, and you have this compound here, not the compound, with the solid phase with your base 1, and this is your base 2, plus you have the solid with the base 1. I should draw. This is base 1 and your free hydroxyl here, this whole thing, which did not react, unreacted this. This is your CpG, unreacted this. Now, you have to make this inert, otherwise it will react in the next phase. So, first thing what we do is we wash away all the liquid out and dry it. So, all these unreacted of the starting material, this is gone, all the byproduct that is in liquid form are all gone, tetrazole all gone, all in liquid phase are gone. What will remain? Only this and this, the unreacted one. Now, we need to cap this, we need to make this inactive. So, that is called the capping stage. So, this is will be capped. It is basically a esterification reaction, you will form a ester here. Once you form the ester, it is very unreactive, it will be dead, it will not react in future steps. So, all you have now is this with your base 1 and base 2. Now, if we want to stop here, also what are the other steps that are remaining, if you want to stop right here. If you want to continue, you can move on just like the 2 steps that I have just described. So, now here as I was saying that it has a +3 oxidation state. So, it needs a oxidation, which is done usually by iodine in presence of water and pyridine. All it does, it includes a double bonded oxygen in this position. This is base 1, this is base 2. OR and this OR means the protection groups. So, you have basically 5 plus 5 oxidation state phosphate ready. You have still have this tail. So, now then once you complete all your synthesis, what is your aim is to get rid of the solid phase, because you want your DNA, not the solid. So, that is done by hydrolysis with an ammonia water. If you just hydrolyze in ammonia water, then this ester bond will cleave and that will give you free from the resin. You will get a free hydroxyl group and your resin will be eliminated out. This is now in solution phase. This can now come in solution phase, solution, and your solid is out. You can take your solution and then can isolate from there. You can dry the ammonia out, dry the water out, you have your compound. And this group as well as this group can be eliminated to get the pure phosphate if you just treat this with ammonium hydroxide at 55 degrees Celsius for few hours. That will give you this. At the same time, so one thing I forgot to mention, that when you use the nucleobases, for example, the guanine or adenine, those have free amine groups which may react. So, that needs a protections. I have talked about the protections of nucleobases already. So, you have to use at the beginning the protecting groups of the nucleobases, base 1 or base 2, if they have free amines. And at the same experimental condition, those protection groups will also be deprotected and you will get the free form of the nucleobases. So, that is how you get a dinucleotide and you can carry on from this stage itself, you can carry on if you want multiple numbers, and that is how we can go really far. At this point 30 to 40 nucleobase pairs are very easy to synthesize, we can go even beyond 50. So, that is how nowadays the DNA is synthesized, and they are called the oligonucleotides and they are used in for various purpose. You will see there are huge applications just in the next module. So, this is the again the cycle, you can go around the cycle again and again and will give you this. So, here is the time frame, how much time you need do you need to complete this. You see it is really fast, really beautiful chemistry, really fascinating chemistry, and every step we have washed to remove all the liquids out. So, first step is washing with the acetonitrile, that takes only just 30 seconds. Detitration, that is the removal of the DMT, just take 50 seconds. Then wash with acetonitrile again, that takes another 30 seconds. Then you make your solid phase dry, because so that no other liquid stays in, that takes another 10 seconds with a pure argon. And then coupling, where you attach the new nucleobase with this, that takes only 30 seconds, maximum 1 minute, 30 seconds to 1 minute per base coupling. And then wash, argon, flush, capping takes another 30 seconds, all the washes takes 30 seconds, oxidation needs a little bit longer time, 45 seconds, and then 30 seconds wash and flush. So, within 5 minutes your reaction is done, full one nucleotide synthesis is done in 5 minutes. So, in 1 hour you can synthesize quite a good length of your DNA, and all automated inside the machine. So, yeah. So, that is what is called the phosphoramidite DNA synthesis, automated solid phase nucleotide synthesis. And with this I will complete the module 2. So, what we have learnt in this module is that I have shown the organic synthesis of pyrimidine and purine nucleobases. We have seen synthetic methods, different methods that we can use or develop for the nucleobase analogues. If you want to synthesize the derivatives of the nucleobases which have other applications apart from the base pairing, for example, in drugs industry and medicinal in medicine industry as pharmaceutical drugs. Then we have also seen how prebiotic chemistry can be done, or can be some ways can be speculated how the evolution of the nucleobases happen when there was no life on earth. Then we have seen the organic synthesis of the nucleosides, how can you couple the nucleobase with the sugar both in RNA or in DNA, and then finally we have seen how a full length DNA, or not a full length, we call it full length DNA because that is how we understand it. So, oligonucleotide, how can you synthesize a oligonucleotide using automated solid phase synthesis. So, that is all for the module 2, and the next lecture I will start the module 3. Thank you.